The formation of the trophectoderm is the first differentiation event that happens in the mammalian embryo. This makes sense since these cells allow the embryo to implant into the uterus. Recall that the trophectoderm or trophoblast cells will give rise to the embryonic portion of the placenta, which is known as the chorion. The chorion not only forms the embryonic portion of the placenta, but also induces the formation of the decidua, which is the maternal portion of the placenta. Additionally, the cells of the chorion also secrete hormones that prevent the immune rejection of the embryo. The chorion is also important for gas and nutrient exchange between the maternal and the fetal blood. The differentiation of cells in the mammalian embryo is influenced by the location of the cells. So most of the descendants of the external cells will give rise to trophectoderm, as depicted in this figure. On the other hand, internal cells are more likely to give rise to the ICM. So these results suggest that the location of the cells can influence the genes that they express and thus influence their fate. As we've seen in previous lectures, this relates to the uneven localization of proteins in these cells. Initially, all the cells of the embryo expressed low levels of OCT4 and CDX2, and these are transcription factors. At this point, a cell can become either ICM or trophoblast. However, as cells divide, asymmetric cell divisions result in the activation of the Hippo pathway in the cells that are internal and the inactivation of that pathway in the superficial cells. The Hippo pathway activates OCT4 expression, and OCT4 is one of the transcription factors that is needed for the maintenance of pluripotency. The outer cells in which the Hippo pathway is inactive express the transcription factor CDX2, and those cells go on to differentiate into trophoblast cells. Once CDX2 becomes expressed in the cells that will give rise to the trophectoderm, it represses OCT4. Similarly, OCT4 represses CDX2 in the ICM. Pluripotency in the ICM is maintained by the expression of the transcription factor OCT4, as well as by the expression of the transcription factors SOX2 and NANOG. These transcription factors bind to the enhancers of their own genes, and they also bind to each other's enhancers. Later on, we'll discuss the events that lead to the eventual inactivation of these transcription factors, as cells lose their pluripotency in their path toward differentiation. Recall that as the embryo moves from the oviduct to the uterus, it undergoes cleavage and organizes itself into a blastocyst. And the cells of the oviduct actually play an active role in the formation of the blastocyst. The progression from the morula stage to the blastocyst stage occurs through a process that is known as cavitation, which simply means the formation of a cavity. Initially, the morula does not have an internal cavity, and that cavity actually forms as a result of the action of sodium pumps in the trophoblast cells, which pump sodium into the interior space of the morula, and that sodium then serves to draw the movement of water by osmosis. The activity of these sodium pumps, and these are sodium potassium and sodium proton pumps, is stimulated by the cells of the oviduct. The water that moves in through osmosis following the sodium influx leads to the formation and the enlargement of the blastocele. In order for the embryo to implant into the uterus, it has to be released from the zona pellucida. If you recall from the lecture on fertilization, the zona pellucida is the extracellular matrix of the mammalian egg. The process through which the embryo leaves the zona is known as hatching, and that involves the release of proteases that partially degrade the zona to create an opening through which the blastocyst can escape. If hatching occurs too early, before the embryo makes it to the uterus, an ectopic pregnancy can result. This figure shows a normal implantation site marked by an X, and the other letters that you see in the figure are locations where the embryo can implant ectopically. The most common location is the oviduct, so tubal ectopic pregnancies are the most common type of ectopic pregnancy. These pregnancies are dangerous because as the embryo grows, it can cause a hemorrhage. While early hatching can lead to an ectopic pregnancy, If the embryo does not hatch from the zona pellucida, the result is instead implantation failure, and that leads to the loss of the pregnancy. Once the embryo hatches from the zona, it will adhere to the epithelial lining of the uterus, which is known as the endometrium. The hormones estrogen and progesterone induce the formation of an extensive ECM, which is secreted by these endometrial cells. This ECM contains collagen, laminin, and fibronectin, among other components, and the trophoblast cells use integrins to bind to that ECM. Peak adherent is also expressed by both the trophoblast cells and by the cells of the endometrium, and it helps to dock the embryo to the uterus. Once the embryo is in contact with the endometrium, the presence of Wnt proteins induce the trophoblast cells to secrete proteases that will help the embryo to bury itself in the uterus. It's evident that the formation of the chorion is essential for mammalian embryonic development, given its roles in nutrient and gas exchange, immune modulation, and implantation. But in addition to the chorion, there are additional extraembryonic membranes, each of which plays a different role. Of these membranes, the amnion and the allantois are derived from the epiblast, which also gives rise to the embryo, and the yolk sac is derived from the primitive endoderm. The amnion functions as a shock absorber, and it also helps to prevent the desiccation of the embryo. The allantois is important for waste collection, but also contributes blood vessels to the placenta. The yolk sac, which is derived from the primitive endoderm, is required for circulatory system development. Interestingly, there are some brain cells, the microglia, which are also derived from the yolk sac. As we'll see, the formation of the yolk sac results from the second differentiation event in the embryo, which is the formation of the primitive endoderm. This figure summarizes the timeline of the events we have described so far in humans. It also shows the origin of the different tissues of the embryo. As you can see, in human embryos, the blastocyst implants between days 5 and 9 of embryonic development. In terms of the origin of the different tissues, recall that the first differentiation event in the mammalian embryo is the formation of the trophoblast or trophectoderm cells. These cells give rise to the chorion. The inner cell mass, which gives rise to the embryo, and the remaining extraembryonic membranes eventually separates into the epiblast, which gives rise to the embryo proper, as well as a few of the embryonic membranes, and the primitive endoderm, which gives rise to the yolk sac. While the formation of the trophectoderm marks the first differentiation event in the mammalian embryo, the separation of the epiblast from the primitive endoderm is the second differentiation event. The cells that became internalized into the blastocyst earlier in development are most likely to remain as pluripotent epiblast, while those that are incorporated later are most likely to give rise to the primitive endoderm. The separation of these two tissues involves a differential expression of the transcription factors NANOG and GATA6. Initially, the blastomeres of the ICM appear, as you see here, as a mosaic, with both GATA6 positive and nano6 positive cells that are intermixed. But these cells separate into the primitive endoderm, which is the GATA6 positive population of cells, and the epiblast, which are NANOG positive cells. It should be noted that this differentiation event is influenced by the presence of fibroblast growth factors. So the cells that received a greater number of fibroblast growth factors go on to give rise to the primitive endoderm.